But today I wanted to go over the structure and function of, in particular, prokaryotic cells. And that's covered in Chapter 4. So when you do the reading, be sure to focus on the part where they're talking about prokaryotic cells, bacteria, and a little bit about archaea. I'm assuming that you already know the basics of the structure of a eukaryotic cell from your general biology. That's why I'm saying emphasize, when you're studying, focus on the prokaryotic cell. But I'm expecting you already know the last half of the chapter about the eukaryotic cell. So if you feel like you need to brush up, go ahead and read that section and do some practice problems. So from Chapter 4, I expect that you will know the shapes and groupings of bacteria, the function of the bacterial cell wall. So this is an additional layer eukaryotic cell, not all eukaryotic cells have, and it's a very important feature of prokaryotes and bacteria in particular. You should know the nomenclature. We already went over the nomenclature, but now that we're learning about the morphology, there's a connection in in the case of many species of bacteria. They're named based on their appearance under the microscope. So you should know the relationship between the morphology and the nomenclature and be able to predict, if you're given the name of a bacterial species, or at least the genus name, you should be able to predict what shape it will look like under the microscope. Structural differences. Okay, so there's several key structural differences. We already started talking about that when we talked about the difference between prokaryotes and eukaryotes in Chapter 1. We will continue to add to the list of differences today. The role of the cell membrane in bacteria, it's important for energy production, so ATP production. So what's in eukaryotic cells, where's the energy powerhouse of the cell? The mitochondria, so that double membraned organelle is very important for ATP production. But in the case of prokaryotes, they don't have these internal organelles, so the cell membrane becomes the site of the proton motive force. Structures beyond the cell membrane, there are several structures that bacteria have. Not all of them have all these structures, but they all have a cell wall for the most part. There are a few exceptions. In addition to the cell wall, they might have an outer membrane, a sticky layer known as the glycocalyx, either a capsule or a slime layer. And they also might have some sort of appendage that helps in motility or attachment. Things like flagella or pili or fimbriae. And we'll go over those today. And you should be familiar with the basic differences in the cell wall structure that distinguish a gram-positive cell from a gram-negative cell. And when we talk about microscopy, we'll talk about a way to directly see that difference. It's an indirect way to see that difference through staining methods. The functions, you should know the functions of appendages. They have a variety of functions, and the DNA structure and how it differs from the eukaryotic DNA structure. Also, there's unique type forms of DNA that are not found in eukaryotic cells that some bacteria have, and those are extra chromosomal pieces of DNA known as plasmids. And then how internal components of the prokaryotes differ from eukaryotes. So they do have some organization, even though they don't have a membrane separating the areas within the cell. They do have an organization to the internal components. Also, you should understand what are endospores and how they protect some bacteria from changes in their environment. And then these are terms that I took from Chapter 4 that you should be familiar with. And then I wanted to start by just giving you an overview of the grand diversity that we see in bacteria. And the diversity starts with differences in morphology, but there are also genetic changes. But originally, when microbiologists began to study these interesting new life forms that had previously been undetectable to us, they first looked at the microscopic morphology and those characteristics. And they also had some ideas of the growth characteristics that differed between these different forms of life. But really, the easiest way for them to study and differentiate was these different species within the domain was by how they looked. And so shape was one of the first things that was used to distinguish and classify these different members of the microbial world that are in the domain bacteria. These are all names of bacteria here. They're the genus name of the different types of bacteria depicted in these electron micrographs here in this slide. And as you can see, they have a variety of shapes, from these long rod-like shape, which is actually, in it, as its name implies, it's called bacillus or bacilli for the plural. There are also more round or shorter rod shapes, like Bordetella. The Bacillus genus is a soil-dwelling bacteria that actually are able to form endospores. Bordetella, things like Bordetella pertussis, that's an example of a pathogen that would cause whooping cough, so a bacterial infection, respiratory infection. There also another type of bacteria that form endospores are Clostridium. They are gut-dwelling bacteria found in humans and animals. And they have actually this unique kind of club shape to their rod-like structure, where one end is a little bit wider than the other end. In the case of the bacillus structure, you can have long rods, you can have short rods, you can have different groupings in pairs or singly. Escherichia coli is an example we already talked about. It's a normal microbiota found in the human gut. It can also be pathogenic. And it also has this bacillus shape. It tends to be shorter when you compare it with other genera like the Bacillus genera. And then so the basic shapes are these rod-like or round shapes like Staphylococcus, which has many, many cells that cluster together. They don't break apart right away after a division, so they make these grape-like clusters that you can see in green here, or Streptococcus, which are a little bit oblong, round, spherical shape. And they tend to group in chains and divide in one plane. So the most basic shapes are the bacilli or the cocci, so the round or the rod shape. But we also have some spiral shape bacteria that take the form of either a special spiral that has an appendage for motility called spirochetes, or simply a coiled shape like spirillum, or also vibrio, which is kind of just a bent, not actually spiraled, but bent rod. So the most common shapes are spherical, or very close to spherical, called coccus when you're talking about a single bacterium, or cocci when you're talking about many. Bacillus, the rod shape, not to be confused with the genus Bacillus, which I just told you about. So the genus Bacillus would always be written with a capital B in italics, so you know it's talking about a type of bacteria and not the shape of the bacteria. Although, as I mentioned, there is a relationship between the name of some types of bacteria and their shape. So right now you can tell that Bacillus, for example, Bacillus subtilis, is a rod shape bacteria. Yes?
Is that the real color of the bacteria?
These are not, no, all of these images are electron micrographs. They come out of the microscope on a film that's black and white. And then using the computer, they've added color back to the image. So it's based on the grayscale image that they get, that the scientist gets from the micrograph. So it looks pretty, but it's not actually the way that the bacteria look. What color are bacteria in general? Anyone know? Okay, I have one person thinks green, white, yellow, translucent. You got it. They're, for the most part, they're colorless. When they grow on as a group or a community of bacteria, you can see different colors. And we'll see that in lab when we grow them on a solid media. We don't have to look through the microscope because there's so many bacterial cells in that one spot on the plate that we can see the color that they form as a community. But when you look at them under the microscope, they're colorless. That's why it's so important to stain them or to visualize them with some indirect method, either by using the light, the refraction of light, as we talked about with some of the we will be talking about when we talk about microscopy, or with a beam of electrons, and which electrons bounce off the grid or go through the grid will determine how the image comes out in the electron micrograph. Okay, so other unusual shapes do exist among different species of bacteria. They include a rather round or short rod called a coccobacillus, and it has this appearance where it's a very short, it looks like a very short rod. It's a little hard to distinguish under the microscope unless you have a really powerful magnification like it with the electron microscope, but may appear as cocci when you look at it under the compound microscope like we're using in lab. Vibrio, as I mentioned, it's a rod, but it has a curve in it. Example of that would be Vibrio cholera, the causative agent of cholera. As the name implies, the Vibrio genus, it has the shape of this bent rod, the Vibrio shape. But it would not be written like this. It would be written in italics, and then you would know it's the type of bacteria, not the shape of the bacteria that's being referred to. Spirillum are kind of wavy rods. They sometimes have means of motility, like here you can see in this electron micrograph. This is the bacterium, and at the end, on either end, there are flagella protruding from the cell. But in the case of the spirochete, it also has this a little bit tighter helical shape, but this kind of wavy spiral shape. But they always have a special appendage called an axial filament that helps them to move. It's actually like a flagellum that's been wrapped around the spiral. So it's very closely connected to the cell wall.
What is the name you said? They always have
It's called an axial filament. It's an appendage that's important for motility.
In our last slide, you said that the rod shape is curly. Is that not?
Oh, really?
And it says under domain bacteria, not in the, sorry, not in this one, in the last class. Under the domain bacteria, it says rod shape, and then it says curly in parentheses.
Oh, coli is actually like Escherichia coli. That's referring to the gut. It's not referring to the shape. Yep. So cocci is the round shape, but not to be confused with other terms like coli, which refers to the colon, actually, that part of the gut where the E. coli is part of the natural, normal microbiota. So there's always exceptions to the rule. While the cell wall is what determines and fixes the shape of bacteria that have these shapes I've already mentioned, there are some bacteria that either don't have a cell wall, so they don't have a fixed shape, or they change shape. And those are called pleomorphic in their shape. In addition to the shape, which is determined by the cell wall, the way that the bacteria divide will determine the grouping. So division can occur in just one plane. So think of a graph, like the x and y axis, and then the z axis comes out of the wall, or makes that third dimension to the space. So if they divide just along one plane, they form chains if they don't break apart. So sometimes the cell will divide, it will form two cells, and it will break apart, and under the microscope you will see single cells. Other times they will stay in pairs, and then when each of these cells divide, it will break apart. So you'll have a mix maybe of pairs and single cells in the when you look at it under the microscope. And still other types of bacteria divide without breaking apart the cells. So they form a complete separation of the cell wall. They are completely two separate cells, but they simply don't break fully apart upon completion of division. And these are chains, which are, in Latin, the name for chain is strept. And so, for example, streptococci would be chains of spherical shaped cells. The name of the species Streptococcus implies that. There can also be specific numbers in the chains, so chains of 4 and chains of 8 would be called tetrads and sarcinae, respectively. But in addition to division in a single plane, and the same is true for the rod shape. So you can have just bacilli, single cells that are rod-shaped, diplobacilli, pairs of rods, or streptobacilli. But in addition to division in one plane, some species of bacteria divide in more than one plane. And if they divide in perpendicular planes, then they form this kind of growing cube of cells. The sarcina is an example of that. And then in the case of if the planes are random and not perpendicular, so not at an exact 90-degree angle, then it tends to look more like a cluster. And Staphylococcus is an example of that, which translates as grape-like spherical cells. So based on these morphological characteristics of both shape and grouping. These were some of the first characteristics that were looked at and used to categorize these different species of bacteria. Of course, now we know a lot more about the bacteria based on their genetic makeup. Okay, so in addition to their microscopic morphology, there's the morphology or the appearance of bacteria and how they grow. So most bacteria, when they're in solution, they will grow in suspension. That means they'll kind of grow individually. But some bacteria have special layers that assist them in adhering to a solid surface. We'll talk about some of those today. And in that way, they're able to form what's seen naturally in the environment and their growth in a community as a biofilm. So this is kind of a slimy layer of bacteria. It doesn't have to be a single species. It's usually a combination of different species that are able to grow in a community together. Some of the products from one species may be utilized as a resource by another species. It's a very organized form of growth that includes many layers. The initial species may be important in establishing that attachment to the solid surface, whereas other types of bacteria in the community may not be able to function as those initial, as the types that attach initially onto the surface, but they can become part of the community after those initial species make that really close and strong adherent contact with a solid surface. So this is how we see bacteria growing in nature. If you've ever stuck your finger down your kitchen drain or your bathtub drain, you've touched a biofilm. But in addition to where we find them in nature or in our homes, these types of growth in communities known as biofilms are medically relevant because they are much, much more difficult to treat than a bacterial infection of a type of bacteria that's not able to form a biofilm. So why do you think it would be more difficult to treat a community of bacteria that are in this close-knit community versus bacteria, for example, that are growing individually?
Well, I guess first because they're protected, and second because when they reproduce them.
Okay, so yes, they're protected. So what do we usually use to treat bacterial infections? Antibiotics. Okay, so if the drug cannot reach those lower layers of the biofilm, those bacteria are protected. They persist and they go on growing. And also, while some of them may not be protected, the most outermost exposed bacteria, there's always more below to persist. That's the same is true not just for the antibiotics that we use to treat these infections, but also for our immune system. Our immune system has trouble detecting bacteria that are hidden by other bacteria. They will only detect the ones on the outside exposed surface of this community. So our immune response is also hindered by the formation of biofilms. So these biofilms have to form on a surface. Usually most of our body surfaces are can have a continuous turnover. So the cells of our skin, for example, are constantly being sloughed off. We're losing a lot of the bacteria that's part of our normal microbiota all day long. But in the case of things like medical devices like catheters, that are inserted for a temporary time into our tissues, or even permanent devices like an artificial hip, for example. The bacteria can form biofilms on these surfaces and cause infection in that way. The other type of multicellular association that some bacteria are able to form is a swarm or a community of cells, and that's typical of the myxobacteria genera of bacteria. So these bacteria actually are only able to persist as a group of organisms. And they have special enzymes that they release in order to degrade the material around them so that they can move as a community throughout the environment. But they require nutrients just like all bacteria, and so in the absence of sufficient nutrients or water, these communities can form a more dormant form of life called fruiting bodies. So they no longer require as many nutrients or as much water, and they can kind of sit around in this fruiting body until the return of those resources.